So hi everyone, I'm Megan and I'm a PhD student at the Roger Williams Institute of Hepatology at King's College London. So today I'm going to present our work, which is transcriptomic profiling identifies ribosomal proteins as novel genes of interest in primary sclerosing cholangitis associated cholangiocarcinoma. Primary sclerosing cholangitis is a chronic, rare and complex liver disease which primarily affects the bile ducts, and its hallmark characteristics include inflammation, fibrosis and stricturing. Currently, PSC lacks treatment options, but PSC is one of the most well-recognized Western risk factors for cholangiocarcinoma. Cholangiocarcinoma is a very common complication for PSC patients. It is the most common malignancy seen in PSC. But currently, there is a very limited body of literature surrounding PSC-associated CCA. And so currently the etiopathogenesis of PSC-associated CCA is not clear. Very few omic studies exist which focus on highlighting the genetic, transcriptomic or proteomic differences underpinning PSC-associated CCA. We currently have problems with diagnosing CCA and PSC as clinically the two look very similar. Number of biomarkers we have which we can use to identify CCA and PSC are limited. And furthermore, the stratification of PSC patients, which are most at risk of CCA, is not possible. Finally, the degree of similarity between PSC-associated CCA and CCA is not very clear. And thus, whether therapeutic strategies applied to CCA will be also applicable to PSC-associated CCA is not known. So my project aims to provide novel insight into the differential gene expression in PSC-associated CCA by undertaking massive analysis of cDNA-end or MACE sequencing. So MACE sequencing is a RNA sequencing technique which has been optimized for formalin-fixed and paraffin-embedded tissue samples. RNA sequencing technique to the 3' end. So firstly, we had to identify our FFPE samples, our patient samples. We identified 9 PSC samples, 10 PSC-associated CCA, and 10 CCA samples. The FFPE box were serially sectioned. We then identified the region of interest on the glass slide, and then we scraped that region of interest off. The RNA was extracted, cDNA synthesized, library prepared, and then the Illumina sequencing was done. And then finally, the bioinformatic pipeline started with quality control, then read mapping or an alignment, quantification, and differential expression analysis. So this was done by GenXPro in Germany. For our results, firstly, as you can see here in the principal component analysis, which shows us the similarity of our samples based on clustering. As you can see in the pink, the PSC bile ducts formed quite a distinct separate group. In the blues and red, you can see the PSC background liver, the PSC CCA background liver, and the background liver. These form a cluster, but there is some separation between the different background livers. In the green and brown, you can see the CCA tumor and the PSC CCA tumor. Again, although clustering together, there is some distinction between the CCA tumor and the PSC-associated CCA tumor. There were 3 main comparisons of interest. This was PSC CCA to CCA. So we compared these in order to identify any differences, any different genes which may be involved in the development differentially of PSC-associated CCA and CCA. We then compared PSC-associated CCA to PSC bile ducts, and this was in order to identify any genes which may be involved in the development of CCA and PSC. And then finally, we compared PSC-CCA surrounding liver to PSC surrounding liver. And this comparison was done again to identify any genes which may be involved in the development of CCA and PSC to see if the background liver is involved in the development of CCA. So as you can see, we put these genes into David to look at the biological processes most significantly up or down regulated. Most significantly upregulated in PSC-CCA when compared to CCA included ion transport and synaptic signaling. Most significantly downregulated in PSC-CCA included protein targeting to the ER. When we then compared PSC-CCA to PSC bile ducts, most upregulated was cell adhesion, and again downregulated, we saw protein targeting to the ER. The PSC-CCA surrounding liver to PSC surrounding liver comparison, we found that regulation of intracellular signaling transduction was upregulated, and protein targeting to the ER was downregulated. As you can imagine, this analysis provided quite a vast quantity of data with a very large number of differentially expressed genes. So in order to identify some genes of interest, which we could pursue further to try and identify genes which are involved in the development of cholangiocarcinoma in primary sclerosing cholangitis, we looked specifically firstly at the protein coding genes. We then broke these down and we looked at them based on whether they were up or down regulated. And we looked, as I just mentioned, at the biological processes most significantly up or down regulated in PSC-associated CCA when compared to CCA not on a PSC background. Next was we looked at the top 10 biological processes from David, and we compared those to gProfiler and GCA. We then cross-referenced with the PSC-CCA versus PSC bile duct comparison. Now, we did this in order to identify genes which were significantly higher or lower in PSC-associated CCA when compared to both the PSC bile ducts and CCA tumour. And that told us which genes were unique to the development of PSC-associated CCA. If the gene was downregulated in PSC-CCA, we then identified the most significant and pursued those of genes of interest. Whereas if they were upregulated in PSC-CCA, we looked at the significance, and then we also looked at a threshold count, which we set at 2 raw counts. If these were above this, they were again pursued as genes of interest. Looked at those biological processes, as you can see. So following this flowchart, we identified the ribosomal processes were the most significantly downregulated in PSC-CCA when compared to PSC bile ducts and CCA. We put those genes into string DB to look at whether those genes would be predicting, whether their proteins would be predicted to interact. And as you can see here, this ribosomal processes is largely downregulated. So all the genes are predicted largely to interact, and this whole process is downregulated. And for the ion transport, so these were the genes which were significantly higher in PSC-CCA when compared to CCA, and then also the PSC bile ducts. And as you can see in point 5, these genes largely, the proteins largely are predicted not to interact. So here this represents a biological process which is upregulated, but perhaps a number of different pathways. What conclusions can be drawn from this? Well, this transcriptomic data set identifies some novel differences between PSC-associated CCA and CCA not on a PSC background. So ribosomal processes are predicted to be downregulated and ion transport upregulated. And how exactly these identified genes of interest play a role in the development of CCA and PSC is not clear, but it does warrant further investigation. So just some acknowledgements. Thank you to the AMMF Cholangiocarcinoma Charity for choosing this work to be presented at your amazing conference. Thank you to the King's College Hospital Charity for funding this work, and everyone at the Foundation for Liver Research for their support.